So guys, this is the structure of Lac Operon, and we will be studying the gene expression of this Operon system, right? So let's first see what is basically a Lac Operon. A Lac Operon or Operon system is a sequence of gene that is regulated for the lactose consumption or lactose metabolism, and that lactose metabolism is dependent basically on three types of enzyme. Number one is beta galactosidase, number two is permease, and number three is transacetylase. Now these enzymes should be in a manner that in which we can find that particularly in this section these enzymes should be in certain amount, but when will these enzymes operate when we will not need lactose or the glucose level rises higher or galactose level rises high, then what kind of mechanism, how does the total system turns off or turns on? We will see that in this particular section. So first let's look at the structure of that Lac Operon. In this case, first we will get a I gene or repressor gene. It will produce a repressor protein. Repressor mRNA will be produced, and that mRNA will produce a repressor protein. So it is kind of a protein that has a binding site of lactose, right? Now in this case we have another one, that is the RNA polymerase binding site. In this particular section the RNA polymerase, RNA pol will bind here, RNA pol 1 takes place, and then we will have a operator sequence in which the proteins and that particular this protein will sit in this operator sequence when lactose is absent or lower in the system, and then we will have a beta galactosidase. Beta galactosidase, what does it do? Actually it breaks down lactose into galactose and glucose, right? Then we have another enzyme, permease, which takes place in the cell membrane. In this particular cell membrane we will have number of permeases. Whenever this system is turned on and permease enzyme is produced, it will increase in number in the cell membranes. And then another enzyme called transacetylase, it will convert the transacetyl compounds into a different section. That's a particularly on off switch. So we in this particular section we will go for these two gene regulations in here. So okay, so let's move on to our structure and let's see what happens when too much lactose is consumed, and then what kind of gene regulation is observed in this case. Now when there is no lactose in a particular cell or less amount of lactose is in a particular cell, there will be some amount of permease which will allow that certain amount of lactose to come in. So lactose will be coming in via this permease, right? Now in this case I have told you that this one will be having an mRNA which will produce a repressor protein repressor protein, and that repressor protein will sit on this position. In absence of lactose, what would happen? Another gene regulation will show a particular movement towards this gene sequence, and it will produce the required mRNA for the repressor protein, and that repressor protein will bind to this operator sequence. So this is the RP, right, repressor protein. And afterwards, whenever so no transcription will take place in this section. But what happens when lactose starts to enter via cell, via the permease into a particular cell? Let's see what happens in this case. What we have seen earlier, that in this particular section the repressor protein was sitting, and when from the via permease the lactose molecules come inside the cell, these lactose molecules will combine. These are lactose, right? Lactose molecule will combine and form a tetramer. These are known as allolactose, which will be known as allolactose. So these lactose will be coming to the cell. Now what does this allolactose do? First, in this case, this particular repressor enzyme will accept the allolactose in the tetrameric conditions. The lactose will bind in this section, and this is the step 1. And in step 2, the repressor protein will be released from this section along with the lactose molecule, right? So if the repressor protein is released from here, then no repressor protein will be present in this section further. And now if the RNA polymerase one comes to this section, and the transcription starts, right? And the transcription starts. Now what happens when that particular transcription starts? First, I hope you have write it down all. Then whenever there are lactose molecules. Now what happens in this particular section? The RNA polymerase RNA pol has bound to this section, and then afterwards the operator sequence remains empty, right? The operator sequences remain empty and the transcription has started. So the Z sequence will produce but beta galactosidase, and that beta galactosidase will produce glucose and galactose in too much ways, right? And afterwards the permease enzyme will be produced. These are permease, right? These enzymes are permease. What would happen if this is a cell membrane? That cell membrane will get lots and lots of permease, right? And so the entry of the lactose lots and lots of permeases. So the entry of the lactose will become higher, and whenever the amount of glucose and galactose is increased, it will start the lactose, it will signal the repressor gene, this one, repressor gene, to produce the repressor protein. The repressor protein will be forming again. Whenever there is adequate amount of lactose in a particular cell, then what would happen? The repressor protein will come again. Then what we will be able to see, that repressor protein in this section will come again, right, and it will start to produce the mRNA, and mRNA will produce the repressor protein, which will sit in the operator sequence, and then the number of lactose will try to hold this position, and again no transcription will take place. Okay, so first again it will not produce any kind of enzyme, right? So this was the all about our gene regulation, okay, of lac operon. So every kind of gene has a turn on and turn off switch, and that is why we need that turn on and turn off switch for controlling a particular gene sequence and controlling a particular protein structure to develop, right? Okay, so thank you folks. So guys, let's recap the Operon system again. Now there are polycistronic genes, polycistronic genes are there, and monocistronic genes are there, right? So in case of polycistronic, we can find it in case of E. coli, right? So we will be observing the lac operon system in case of the polycistronic operon system, right? Okay, so let's recap. First, in the absence, when there is no lactose present, what happens? The repressor I gene produces one mRNA that transcribes a particular protein known as the repressor protein. This is known as the repressor protein, right? A repressor protein typically has a lactose binding site. This has a lactose binding site, right? So when there is no lactose in the cell, what would happen? The lactose binding site will not appear, and this one will be sitting in the operator region. So operator region. Now what happens? Whenever the RNA pol comes in this place, RNA pol, it cannot move further, so no transcription, it cannot. कैसे जाएगा? क्योंकि यहाँ पे एक blockage है, right? So these blockage due to that repressor protein will result in no transcription. So none of the enzymes of beta galactosidase, permease or transacetylase will be formed, right? Now let's see what happens in case of lactose. Suppose this is a cell membrane, this is our gene. The I repressor protein that was produced early, when lactose is entering in this particular section, what would happen? It will form a tetramer, known as allolactose. And now this allolactose will be sitting in this position, right? So if it is bound in this section, the allolactose will bind in here, and it will release the repressor protein along with the lactose molecule. So it will be gone for that timing, right? Now if the RNA pol comes here, then the transcription will start again. So this will switch on the mechanism, right? And both beta galactosidase, permease and trans-acetylase will start to form again. And what would happen? The permease will increase the number of entry points in the particular cell. Whenever too many permeases are there, it will open too many gates and too many lactose molecule will come in this section, and what would happen? It will start to digest the lactose to form glucose and galactose, and that glucose will be consumed to produce ATP. So that was the whole Lac Operon structure, and how the system is switched off whenever the glucose level increases in much higher position. The permease will shut off the entry of this lactose molecule, and that lactose molecule will go away, and the repressor protein that was produced earlier will sit here again. Okay, in presence of lactose, these things happen. So that was all from our Lac Operon system. So the chapter is completed and you have to do the homework right now. Okay, thank you very much. Please subscribe and tell us to do whatever you want, or what kind of video you want in regarding this biology chapter, right? Okay, thank you folks.